Microbiology. A2 Biology. Terminology revision. Pathogens are microorganisms that cause disease. Capsid is the protein coat of a virus. Capsomer is the repeating protein units of a capsid. VAPS are virus attachment particles, specific protein antigens, that target host cell membranes. Envelope, the coat around the outside of a virus, is composed of lipids from the host cell. DNA, RNA viruses are viruses that have DNA, RNA genetic material. Retrovirus are RNA viruses, which control viral DNA production by inserting into the host DNA. Reverse transcriptase, an enzyme synthesized during the retrovirus life cycle, produces DNA from viral RNA. Pili are the thread-like projections on the surface of the cell. Flagella, a rotating whip-like structure which allows bacteria to move. Capsule, a protective polysaccharide layer outside the cell wall. Mesosomes are the internal extensions or folds of the membrane. These may be sites of cellular respiration. Plasmids are small circular pieces of DNA. Vectors transmits pathogens from one host to another. Provirus, the DNA inserted into the host cell during the lysogenic pathway. Latent, the non-virulent state of a virus in a host cell. Lysogeny, a period when a virus is part of the reproducing host cell but does not adversely affect it. Bacteriophage, a type of virus which infects bacteria. Exocytosis, when a vesicle fuses with the cell membrane and its contents are released outside the cell. Lysis, cell death by rupture of cell wall or cell membrane. Binary fission is asexual reproduction. A bacteria divides into two. Generation time, the doubling time of bacterial populations, the time between bacterial divisions. Lag phase, when the bacteria are adapting to new environment, not reproducing at maximum rate. Log, exponential phase, the bacterial reproduction rate is at or near maximum. Stationary phase, the total growth rate is zero, the rate of reproduction is equal to the rate of death. Death phase, reproduction has almost completely stopped, the rate of death is increasing. Sterile, free from bacteria or other living organisms. Nutrient or culture medium, a substance used to grow microorganisms, can be liquid or solid. Nutrient broth, a liquid culture medium. Nutrient agar, a jelly extracted from seaweed, a solid culture medium. Inoculation, the process of transferring microorganisms to a culture medium. Hemocytometer, a microscope slide with lines, is used for counting cells. Turbidity, a method for measuring concentration, measures the amount of light passing through. Turbid, something opaque due to suspended material in the solution. Dilution plating, a method used to count bacterial colonies on culture plates. Total viable cell count, a measure of the number of living cells in a specific volume of culture. Communicable, pathogenic diseases which can be passed from one host to another. Bacterial flora, a combination of different bacterial species in or on part of the body. Sebum, an oily substance produced by the skin, it inhibits microorganism growth. Lysozymes are enzymes which destroy bacteria, found in tears and other body secretions. Endotoxins, lipopolysaccharides, the outer layer of gram-negative bacterial cell walls are toxic to other cells. Exotoxins, soluble proteins produced by bacteria which act as toxins. Primary infection, initial stage of tuberculosis, multiplying in the lungs with no obvious signs of infection. Tubercle, a localized mass of tissue, made of dead bacteria and macrophages. AIDS, acquired immunodeficiency syndrome, causes destruction of T helper cells. HIV, human immunodeficiency virus, a retrovirus that causes AIDS. HIV positive, has HIV-specific antibodies present in the blood. Antiretroviral drugs, drugs used against retroviruses. Don't forget to like and subscribe.